Hello friends, this video on Principles of Inheritance Part 42 is brought to you by examfear.com. No more fear from exam. Let us look at the next Mendelian disorder, that is sickle cell anemia. So let us see what happens in sickle cell anemia. So before we talk about sickle cell anemia, let us first understand about hemoglobin because it has a lot to do with hemoglobin. I hope all of you know what is anemia. So whenever you have lack of hemoglobin in your blood, you say, people say that okay, this person is suffering from anemia. So lack of hemoglobin in blood is termed as anemia. Now this is a special type of anemia where the red blood cells get a change in their shape. Now before that, what does hemoglobin do? Hemoglobin is nothing but a protein that is present in the red blood cells and it helps in the transport of oxygen. So it has the capacity. So hemoglobin has iron and this iron has the capability to combine with oxygen, and that is how it carries oxygen from one part of the body to the other. So it is extremely important because blood serves a very important purpose of carrying oxygen from one part of the body to another. Now there is a gene that codes for hemoglobin is present, right? And if this gene gets mutated, if there is some change in that particular gene, what will happen? The hemoglobin synthesis will be affected. Now when hemoglobin synthesis gets affected, oxygen transportation also gets affected, right? So in sickle cell anemia, the RBCs become sickle shaped and that is why they are called sickle cell anemia. Normally if you look at the shape of RBC, the red blood cells, normally they are rounded structures, somewhat like this. But when somebody is suffering from sickle cell anemia, the shape changes to a shape like this. And because of this particular shape of the RBCs, this disease is known as sickle cell anemia. Now let us try to understand how this gene actually affects the hemoglobin production. So let us assume that the gene that controls the hemoglobin. So there will be a gene that controls the hemoglobin. Correct? Now, where is this gene located? The gene is located on the chromosome. And what is the gene composed of? It is nothing but the DNAs. So the DNAs have the sequence of bases. Let us suppose, just for example, let us take any sequence. Let us suppose this is how the sequence looks like. The sequence on the gene, this is how it looks like. Now, when this gene send instruction for protein synthesis, what happens? First of all, a messenger RNA is being produced, that is known as RNA transcription. So from the sequence of the DNA, RNA is coded. So what type of RNA is produced here? Let us suppose this is how RNA is being produced, and then this RNA helps in protein synthesis because RNA sends the signal to ribosomes, and the ribosomes are the sites for protein synthesis. Right? So proteins are all made up of amino acids. So what are the amino acids that are being produced by this RNA? Let us suppose this particular section of RNA produces an amino acid called proline. Again, let us suppose this particular section produces another amino acid called glutamic acid. Again, the rest of the portion, this produces another amino acid called glutamic acid. So this is how the amino acids are being synthesized, and these amino acids form the protein, and this gives a shape like this, of a spherical kind of a shape to the red blood cells. So this is how the normal synthesis take place. Correct? Now let us suppose that this gene gets mutated somewhere. So mutated in the sense, mutation can happen, maybe one of this got replaced, that is one of these values got substituted, or one of them has been removed, or one extra base has been added. So anything can happen. So what happens in sickle cell anemia? So when somebody is suffering from sickle cell anemia, that means what has actually happened is that the sequence on the DNA has changed. Earlier this was the sequence, right? So now let us suppose the sequence has changed. Let us suppose instead of T, it is now A. So that is the change. So here you can see the change. Now because of this small change, what happens? The messenger RNA that is being coded from this DNA, that also changes. So it becomes, instead of A, it is now U. Here it was A, now it is U. Right? Now when this sequence changes, now this was coding for some specific amino acids. Now earlier it was coding for say glutamic acid. Now what is happening? The first part is still coding for proline because there is no change, but the second part has changed. So instead of glutamic acid, what is happening is another amino acid called valine is being produced, and the remaining part is the same. Now here, due to the change of this amino acid, a different protein has been synthesized altogether, right? Now when the structure of the protein changes, its function also changes. That is what we have studied about proteins, right? The tertiary structure or the quaternary structure of a protein tells a lot about its function. Because when you talk about proteins, many of them act as enzymes where they will have to attach themselves to the substrate. Now let us suppose the structure of the protein only changed. So they cannot attach to that specific substrate. So maybe they will attach to some other substrate and something else will be produced. So the same thing happens here. So here instead of glutamic acid, valine amino acid has been formed, and which causes a change in the shape of the RBCs, and that is why the RBCs become sickle shaped, and this disease is termed as sickle cell anemia, because the cells, the red blood cells, are sickle shaped. And anemia because there is lack of hemoglobin, because the gene which control the production of hemoglobin only got mutated. So the hemoglobin production got affected. So this is what happens in sickle cell anemia. Now the question is how pedigree analysis helps in knowing that whether the offspring will be suffering from sickle cell anemia or not. So let us try to look at that. So sickle cell anemia is an autosome linked recessive trait. So this is also recessive. That means a person is affected only when he or she gets it from both the parents. And this is autosome linked. So it is not sex linked. So it is not that it will be different in case of males and it will be different in case of females. That is not the case. So in case of this disease, if somebody is affected, so this is how it will be. So the hemoglobin is denoted by HB. So that if the person is affected with sickle cell anemia, the gene is denoted by HBS. S stands for sickle cell. So if somebody is affected, this is how the genotype would be. That means he has inherited it from both the parents, right? So that means the individual is affected. If somebody has only one gene for sickle cell anemia, the other one is for normal. So for normal one, which we denote it by HbA. So A is for the normal RBCs and S is for the sickle cell RBCs. So if it is HbS, HbA, that means even though the person is not affected, but the person definitely is a carrier. So it can pass it on to the next generation. And if the genotype is HbA and HbA, that means the person is absolutely normal. He is neither affected nor is he a carrier of the sickle cell gene. Right? So these are the three types of genotypes that would be possible in this case. So it is transmitted to the offspring only when both the parents are carrier, and that is why it is a recessive trait. So when HbS and HbA both are present, so HbA is dominant trait and HbS is recessive trait. So let us try to look at its pedigree. So let us take a scenario where we see that the male is a carrier and the female is also a carrier. So if you consider a scenario like this, like how you are seeing it in this pedigree chart. So here what does it show? It shows the red color always shows that somebody is affected. So here if you see it is half red and half blue, that means it is a carrier. This is also half red, half blue, that means this is a carrier. Right? So how do we denote a carrier? This is how a carrier is denoted. So this is a carrier. Right? So both of them are carrier. Now male is a carrier, female is a carrier. So what are the chances of their kids, of their offspring? So if you see, the chances are that half of the males will be normal and half of the females will be carrier. See, here you can see the males. Half of the males will be affected and half of them will be carrier. So here you can see. So if you try to see it this way, you can see. It is suppose if HbS and HbA, if they are crossed with each other. So what happens? There are chances that this can combine with this, or this can combine with this. Again, this can combine with this, or this can combine with this. So there is a possibility that it will be HbS, HbS, or it will be HbS, HbA, or it can be HBS, HBA, or it can be HBA, HBA. So what does this mean? This means a normal individual. This means an affected individual. So there is a possibility that 1/4 of them will be normal, 1/4 of them will be affected, and the remaining 50% of them will be carriers. So this is what you see here. So out of the 4 offsprings, if you see, 25% of them are normal. So here you can see normal ones. Some of them will be affected and some of them will be carrier. So that is how the probability will be. Now again, when each of them get married and then they produce their offspring, so again you likewise do the calculation and you can prepare a pedigree chart like this. Right? So this is how sickle cell anemia can be diagnosed, or it cannot be diagnosed basically, it can be guessed that how the disease can be inherited to the next generations. Now some of the symptoms which are very common in a patient suffering from sickle cell anemia is, as I said, the RBCs become sickle shaped, and as a result the RBCs become very fragile. Because of this, they often have pain in their bones, lungs, abdomen and joints. So this abnormal shape of RBCs also interfere with their function, and therefore they cannot flow properly through the blood vessels. Now when your blood cannot flow properly through the blood vessels, that can actually cause a serious threat to your life at a certain point of time. So that was about sickle cell anemia. So let us look at the next Mendelian disorder, that is phenylketonuria. So let us look at what is phenylketonuria. So the name itself gives a hint what it is all about. So phenylketonuria, it is an inborn error of metabolism of phenylalanine. So what is this phenylalanine? This is a compound which is present inside our body and which needs to be metabolized. And how is it metabolized? It is generally metabolized with the help of an enzyme called hydroxylase, and this enzyme normally converts phenylalanine into another compound called tyrosine, and this tyrosine later forms several catecholamines, which are, you would have heard about the name catecholamines while we were talking about the hormones which are produced by the adrenal gland. So that is how this phenylalanine is utilized or metabolized inside the body. Now, if this enzyme itself is not available, so if this enzyme is not there, what will happen? The metabolism of phenylalanine will not happen. So in that case, what will happen to phenylalanine? So phenylalanine will get accumulated because it is not getting converted into tyrosine. So more and more phenylalanine will get accumulated, and then some of this phenylalanine will get converted into phenylpyruvic acid and some of their derivatives, and these are extremely harmful for the body because these kind of substances can cause mental retardation. Right? So that ways it can actually make a person mentally retarded. And why all this happened? This happened due to the lack of this enzyme, the hydroxylase enzyme. Now, why this enzyme was not there? The lack of this enzyme was because of some genetic disarrangement. Because of this enzyme was not getting properly produced. Right, as I said, protein synthesis, then protein synthesis related to the production of various proteins and enzymes and etc. So in this case also, if you look at how this disease is being inherited from one generation to the next. Now here you can see this is how phenylalanine is. This is phenylalanine and this is phenylpyruvic acid. So this is the structure. So when it is not converted or when it is not acted upon by hydroxylase, this by itself gets converted into phenylpyruvic acid and its derivatives, which can cause mental retardation in individuals. Now, if you talk about how it gets inherited from one generation to the next, it is also autosome linked recessive trait. So the pedigree of phenylketonuria will be similar to that of sickle cell anemia because that was also a recessive trait, this is also a recessive trait. So it will be transmitted to the offspring only when both the parents are carriers. So that is why here I have displayed the same pedigree which we had used for sickle cell anemia. So the transmission is going to be the similar way. Thank you. Please visit www.examfear.com to watch more videos. attempt free online test, get free study material, find tutors and mentors. Thank you once again.